All right, hi there. So we're going to talk about the chromatin immunoprecipitation assay, or ChIP assay. And the goal of this one is to find proteins that are associating or sticking to specific DNA sequences. All right, so with that in mind, most of the proteins that are going to stick to DNA are going to be transcription factors, and transcription factors help to turn on or off genes and make RNA. So that's why we care about it, is we're trying to figure out which genes are turned on or turned off by which proteins, excuse me, and how that affects different conditions in the cell. All right. So a lot of these are, there are a few different concepts that we're going to talk about with the ChIP assay, but we need to talk about a few of these different components individually, and then we'll put them all together. The first one is the ChIP assay, stands for chromatin immunoprecipitation. So the immuno part deals with antibodies. These are proteins. They're usually kind of Y-shaped, and the tips of the Y stick to whatever they're supposed to stick to. So in the case of if one of us is sick with the flu, then the tips of the Y would stick to parts of the flu virus, specifically parts of the protein on the flu virus, and that helps us fight off that infection. So with antibodies, they're very, very specific to individual proteins and even individual small regions on proteins. So that helps us with this assay to make sure we're only detecting the protein that we're interested in. Okay, so now that we know and remember what an antibody is, we already said an antibody is very specific to one region of a protein, and it's only generally good to recognize that one protein. So if we've got a whole bunch of proteins that are inside of a cell and we just burst open that cell and we add in antibodies, then those antibodies are going to stick to whatever single protein that we're interested in that's in that side of that cell. So in this case, the red triangles are the protein that we're interested in. Those antibodies are going to stick to the red triangles. And then you're probably noticing this purple thing. So the antibody in this case is attached to a small bead. It's the bead gives the antibody weight. So then when we spin things down and centrifuge it, the beads are going to stick to the bottom of the tube. The beads are stuck to the antibody. The antibody is stuck to the protein that it's supposed to recognize. And all the other proteins kind of get discarded. So at that point, we're just left with the bead and the antibody and the one protein we're interested in, one kind of protein we're interested in, all at the bottom of the tube. So we detached the protein from the antibody, and then we just purified the protein we were looking for. Okay, so again, we've got an antibody. Now we've got an antibody stuck to a bead, and that antibody is going to recognize a protein. And now, like we said, we want to apply this immunoprecipitation to find specific proteins that are stuck to particular regions on DNA. So we're just going to add on one more step. Rather than start here where the bead attached to the antibody is sticking to the protein, we're going to back up a little bit and say that, okay, now instead of red triangles, now our antibody is specific to purple kind of ovals. The first thing we're going to do is the DNA and the proteins that are interacting with each other, we're going to add a chemical. It's generally formaldehyde, but add a chemical that will make it so the protein is, and all of the proteins that are already associated with the DNA are going to be covalently bound to the DNA. So then what we're going to do is sonicate them. So this uses sound waves, and it's kind of odd, but we can use sound waves to break DNA into very narrow ranges of sizes. So in this case, it would be a few hundred base pairs. So we took the whole chromatin of a chromosome, we exposed it to sound waves that broke it into little pieces. And if you look, each of those proteins that were associated with the DNA, since they're now covalently bound to the DNA, they're going to stay stuck to the DNA. All right, now we go into just the immunoprecipitation part with the antibody and the bead that we already talked about. So we've already got the proteins, and we've already got the proteins stuck to the DNA. So now we add the beads with the antibodies, and the antibodies are going to stick to the proteins that they recognize. That's going to be the purple ovals. And the purple ovals are covalently bound to DNA. So we now have a bead stuck to an antibody, stuck to a protein, stuck to DNA. That's exactly what we want, right? The whole point of this is to find proteins that stick to particular regions in DNA. So what we have to do next is we centrifuge this, and all of the beads with everything that they're stuck to sink to the bottom of the tube. Everything we're not interested in kind of floats away. And then we reverse those cross links. So the proteins are now no longer bound to the DNA. They did their job. We found regions of the DNA that are only associated with those purple circles. So then we purify these DNA fragments and we use PCR to see, was the region we were interested in attached to this protein? Okay, so a lot of people tend to forget that PCR is needed, but if you think about it, since we're using PCR, that means we have to know the gene or the region of DNA that we're looking for. We have to know the sequence of it so we can design a primer that's the reverse complement of the two regions, and then it will amplify, PCR will amplify the region between those two primer binding sites. Okay, so once again, ChIP assay is good for looking if you are trying to find whether a specific protein sticks to a specific DNA sequence. This also means we already talked about specific DNA sequences, and we have to know the sequence of them so we can design primers for PCR to work. Well, we also have to have an idea of which protein we want to look for before we even start, so we can find an antibody against it and attach that antibody to the bead and make sure that the proteins that we want are coming out of the solution too, if they're precipitating out. And then this is the results. So a lot of people get a little bit confused, but there are actually two control lanes in these gels after the PCR, and then one lane that tells us what's going on. So the first one is IgG, is an abbreviation for a specific kind of antibody. In this case, the antibody is not supposed to bind to the protein that we want. It's just a random protein that doesn't stick to anything in the sample that we're trying to look for. So in this case, if it's human, it would not recognize any protein that's human. So in that case, the antibody wouldn't recognize any protein. That protein wouldn't be stuck to any DNA. The DNA wouldn't amplify in PCR, so you will get no band there. So that's a negative control. In the middle lane, what we're doing is we are using complete genomic DNA that hasn't been purified, hasn't been immunoprecipitated. It's just all of the DNA to make sure that, yes, our primers work and they do stick to that DNA and they do amplify what's between it. So this is kind of like a positive control. Okay, so this is the experimental well and lane. So what this is telling us is IP means immunoprecipitation, which means everything in front of it or before it is going to be the name of the protein that we're trying to bring down and pull down out of the solution in the tube. So in this case, the name of the protein is BRCA1, and we're trying to see, did BRCA1 immunoprecipitate this region of DNA? Okay, so BRCA1 is a protein. It plays a role in breast cancer in humans. MAD2 is on this side of the gel. This is going to be the region of the DNA that that protein stuck to. So MAD2 is a region in the protein. It's a gene in the, excuse me, it's a region in the DNA that ultimately codes for a protein. But right now we're just looking at nucleotide positions 116 through 619 in that gene of the DNA. And since there's a band here that says that this region of the gene interacts with this protein, so that protein would be immunoprecipitated with the DNA. That DNA would then be amplified with PCR, and we would get a band there. But if you look closely, that doesn't hold true for all regions of the MAD2 region in the DNA. We do get a little bit of a faint band from positions 599 to 1046. We get another strong band here, no band, yes, a band, and no band. So we can see that this protein doesn't stick everywhere. It doesn't coat the whole region of DNA. It's just sticking to a couple of different places. Again, the negative control should never show a band. The positive control should always show a band. And this one, if there's a band, it says that the protein that's measured at the top or or listed at the top associates with the region of the DNA that's along the right side. And then the very last one, SOX9 is a different gene that we know does not interact with the BRCA1 protein. So we would expect there not to be a band here. So this is kind of like a negative control to make sure the IP worked that the way that we thought it should. Okay, so that's the ChIP assay. Again, it's just using antibodies to find proteins that are stuck to DNA. And then we use PCR, run it out on a gel to see if there's a band that indicates whether this protein interacts with this region of the gene. So we do need to know the protein we're looking for because we need to use an antibody against it, and we need to know the region that we're looking for in the DNA so we can design primers against that and make sure the PCR works. So they do have some high throughput ways of doing ChIP assays. So we're not just looking at one gene and one protein. So they have microarrays. They've moved from microarrays, they call the microarrays ChIP, chromatin immunoprecipitation on a microarray chip. Now they're moved more onto chromatin immunoprecipitation assay with sequencing. So they call it ChIP-seq. We won't really go into that, but in this case, we can still look for one protein, but we can look across the whole genome and see exactly where it sticks. And that's a lot more high throughput, a lot more useful if we're trying to get a better idea of what's really going on with this one protein across the whole genome. So hopefully that's helpful and look forward to the next video. Thanks.